forward on. So we have our amino acids. And we're not gonna be doing anything with these quite yet. It's just so you've seen this list before. With our amino acids, these are the Legos that make our proteins. And we have some that our body, we do need to take in our diets because our body cannot create them for themselves. We have others that we cannot create for wait a minute, Doug, get on the wheel. Sorry, Doug is going in a million areas right now. We have some that you need to take in your diet because you cannot create them, and others that you do not have to take in your diet because your body will take other amino acids, reassemble them into these new structures. So then we were just talking about the basic function of our proteins. We had our structural proteins. And the overall folding and recreation of these proteins. We talked about the 4 layers of the structure. We had our primary structure, which is the amino acids chained together. The secondary is where we're looking for natural bends and kinks because it's much easier to fold and manipulate these proteins where there's already a bend. I think of this folding kind of like, you know, the cord that you have on your computer. And when you're looping it, there's a natural bend in the cord and it's very easy to loop. If you've ever tried to loop it against the bend, it's much more difficult. It's going to kink up on you. The exact same thing is happening here. That's the secondary structure. The tertiary is...
Ms. Huff?
Yes.
Someone in chat wanted to know what it means to ionize.
To ionize? To ionize is to create an ion. So when we're creating an ion, and an ion is an atom that has gained or lost an electron. So if I lose an electron or gain an electron to change the charge, I have ionized and I have become an ion. So ionize just means to become an ion.
Okay, thanks.
And thank you for letting me know, Leanna. Guys, I don't have my chat on. I can only do the chat when I'm at home when I have my multiple screens. Chat takes up way too much room on this tiny little laptop screen. So although I do have my my camera, I have like 3 or 4 of you that I scroll through, I do not have the chat. So if you guys have questions, please, please, please just unmute yourself and let me know. Quaternary is where we have 2. Go ahead.
Sorry. Is the test that we're going to be taking, are we going to have to write it in our own answers, or is it going to be like the last one?
No, it will be true and false multiple choice. There will be there will be no fill in the blank. And Sean, I love your shirt. You're looking very festive today. Is today St. Patrick's Day?
Thank you. Yes, ma'am.
Oh my gosh. I didn't even know. I'm getting punched because I'm not wearing anything green. So yes, you look
pinched, not punched, pinched.
Oh, it's pinched. Okay.
There's nobody punching you.
It's like people coming up just punching me in the face. No, you look very handsome and very festive, Sean. I love it. Here's just a visual reference. I love it. I love the tie. The bow tie is what makes it for me. All right, so here's just a picture representation. We talked about this so much yesterday. Here's our primary where you can see each of these little dots represents an amino acid. We have our natural bends that we then use to fold. And then we take those molecules and bond them together. And those are our 4 structures. We had our structural proteins, which are just that. They are forming things. We had our collagen, which is a connective tissue. Probably the most common example of our collagen is the connective tissue that connects our skin to our muscles. As you grow older, I am sad to say, all of you are fetuses, so you don't have to worry about any of this yet. But gravity is kind of a crap fest. Because what's happening is gravity is pulling on your face and your skin all of the time. And the problem is that your skin only has a finite amount of elasticity. If you take a rubber band and you over time stretch it, what happens to it? It loses the elasticity. Same thing is happening here in the skin, in the collagen. We have keratin, functional protein, or the structural protein rather, not functional, structural, that we see it in our specialized, like our claws, our horns, our antlers. And then yesterday we talked about actin and myosin, which are two proteins that interlace and contract and cause your muscles to be able to contract and move. Then we had our functionals. We talked yesterday about hormones and how hormones govern everything about your body. We even have hormones that govern other hormones. It's like this whole multi-level marketing system, MLM. It's the whatever you want to think of, where you have one governing another, governing a body system. And so everything about your body, it's not just estrogen and testosterone, although yes, those are two important reproductive hormones. There are so many hormones, and we're going to talk about them specifically when we get into the endocrine system. But right now, just know that hormones govern everything about you.
Is there
Antibodies.
anything particular that we need to know? Oh, I'm sorry.
No, go ahead.
Is there anything we need to know in particular about the difference between hormones and protein-based hormones?
No.
Okay.
No, hard no. You're
Okay.
obviously someone who has taken some sort of organic chemistry class prior to. No, hard no. I want you to know the difference between a structural and a functional. That's it. Antibodies, remember, is the wanted poster. We talked yesterday a little bit about the COVID vaccine and what an exciting time for medical research, where not only is it giving us the instructions of how to get rid of or to defeat the COVID virus. It's giving us the test run and giving us the instruction manual. That's why we call it a RNA recombinant vaccine. It's really pretty cool. It's pretty cool. Then you have your protein-based hormones. I am not going to ask you the difference, just know that there are. And then we have enzymes. And remember, enzymes we compared to a lock and a key, where they are turning things on and off. They are the light switches of the body, and they are very specific. For example, the light switch here in my office only works the lights in my office. I can't flick that switch and turn anything else on or off. That's the same thing that's happening with these enzymes in your body. They are very, very specific for the thing that they turn on and off. So there's our enzymes. They speed up or initiate chemical responses. I just talked about all of that. Here's our lock and key. Just like I think yesterday, I also kind of, instead of the light switch in my office, I compared it to the key to my car, and that I can go to every other car in the parking lot, but this key is not going to initiate any kind of response except for my own car. That's an enzyme. Then we talked a little bit about nucleic acids. These are the proteins that make up your DNA and your RNA. And what your DNA is the blueprint, the how-to manual for every function in your body. It tells your body not only how to make other things, but why do we need to make them. It codes everything about you. Everything that is nature-based, if you're going into more of a psychology, nature versus nurture, anything that is nature-based, DNA. And you've gotten that from all of your ancestors since the beginning of the dawn of man. We had 5 nucleotides. Nucleotides, remember, are the proteins that bond together to make up DNA. Now in DNA, we have these 4: adenine, guanine, cytosine, and thiamine. And remember, I did say that might be a great multiple choice question. Which of the following is only found in RNA, or which of the following is only in DNA and not RNA? Just saying, I don't know. Might be a question on the test. Anyway, so we have specific nucleotides that bond with only another specific nucleotide. Somebody tell me what adenine bonds to in DNA.
Thymine.
Very
Got it.
nice, apple tree. And then obviously by reason of elimination, put your car in the garage, cytosine and guanine. Now in RNA, adenine is gonna bind with
Uracil.
uracil, yep. So these repeats, okay, because there's only the 4 nucleotides, so we have a limited way of combining them. So what all of your characteristics are dependent on are the order that they are found in the DNA. And if I change the order, suddenly now what we're looking at are different physical traits. So when we're looking at our dog breeds, because dogs have the most slippery physical properties of any animal on the planet, you don't see the kind of diversity in any other animal than you do in your dogs. We have our dolichocephalic, brachiocephalic, you have your prick-eared dogs, you have your pendulous-eared dogs, you have your short legs, long body, you have thin body, long legs, you have the giant breeds like the Wolfhound or the Great Dane. You have your teacup miniature, they're actually called micro breeds now, like your teacup Yorkie, your Silkies. You have different behavioral traits that are genetic. Terriers are gonna dig and bark and kill stuff. Salukis, your sight hounds, are going to be made for speed. Your scent hounds are going to get the blinders on and they're going to follow blindly any scent they pick up. Those are all genetics that you can't train that stuff. And it all has to do with the order that we find these nucleotides in the DNA. So it's really pretty cool. When we get into some other classes, we're going to talk about what happens when something gets in your body and starts like mixing up your DNA. And that's pretty scary because there are some viruses that'll go in and just start be like, hey, let's just start mixing all this stuff up. And then you're in big trouble. So there's our pairs, adenine and thymine in DNA. And then it would obviously be adenine and uracil in RNA, guanine and cytosine. And the order of them makes up our unique individual code. Now, again, as I said, dogs are the only animal on the planet that have the kind of diversity. Now, yes, do humans look different? Of course we do. We all have a different shaped face. We have different colored eyes. Some of us have curly hair. Some of us have pin straight hair. Some of us, like myself, have this wild, weird, untamed just mop on my head. But ultimately, we all have the exact same traits. We all have 2 eyes, 1 nose, 1 mouth. And a human is always going to look like a human, kind of like a dog or a cow. Oh, I think I froze. A cow is always going to look like a cow. Okay, you're never going to have a cow with a curly pigtail or a cow with like dramatically different ears or a cow with teeny short little legs and a huge giant body. Cow always looks like a cow. Now, are there some genetic differences? Of course, you got your Longhorns, your Brahmas, your Angus, your Brangus, your Charolaines. Those are all the cow breeds I know. I just said them all. Tried to look smart and I blew all of them. So you have all these different cow breeds, but ultimately they all look the same. It's only dogs that have this weird genetic propensity for really, really dramatic physical differences. So there's our structure. So here's a nucleotide. And here's a nucleotide, and they're bonded together. And it's the bonding of these two. One is on this strand. You can see the light blue strand. The one that's bonded is on the dark blue. And when they bond together, that's what causes the strand to wind into this double helix shape that we're also familiar with.
Ms. Huff, I have a
Yes.
question.
Sure.
So like back to the cows, wouldn't they like be considered heifers first and then called a cow once they have like a calf?
Yeah, I was just talking about the species bovine, not the specific sex. You're absolutely right. But I was just talking in general, the species of a bovine. It doesn't matter if it's a bull
Oh,
or a
okay.
heifer or a cow, but breed or sex-wise, you're absolutely right. Yes, they're a heifer before and a cow after. I was just referring to the species.
Miss Huff, I have a question.
Sure.
So I have a cow and actually her legs are shorter than her body. Her body is really big and her legs are really short. Why
Okay.
would that be?
Some sort of genetic abnormality. You had a very small heifer breed with a very large bull, or the vice versa. I mean, can you have some physical diversity? Of course you can. Like humans, you have some humans are, 6'5", 6'6". I'm
Or
like,
colorblind.
or yeah, you have, well, that's a genetic abnormality. I'm talking about genetic norms. But you
Just
are going to
physical.
have
Okay.
some subtle dip, because then you're going into, okay, well, you have some people born without limbs. And so I'm talking about just normal genetics. There are differences between us, right? Obviously, as I said, the color of your hair, the amount of melanin in your skin, the amount of pigmentation in your eyes, your genetic predisposition for certain diseases. That's all genetics. Can there be differences? Of course there can. But ultimately, a human always looks like a human. Whereas a dog, you have some that have the little piggy face. Like, think of a French bulldog. Let's think of two extremes. French bulldog, Irish wolfhound. We don't have that diversity in any other species. So RNA is kind of cool. RNA is how the Lord DNA, the King DNA, I should get one of my tiaras out here. Why? Oh, I've lost all my tiaras. Oh no, someone's come in and stolen my tiaras. Well, that sucks. I have three. I know, I have 3 tiaras. Oh well, I don't really need it. Anyway, you have King DNA, and he's just in his ivory nucleus throne, sending out orders to the peasants. But he's going to be able to get those orders out of the kingdom, out of the castle, the nuclei castle. And so we have RNA. So initially what happens is DNA goes, oh, I'm just going to use an example of, we need to create a protein. Let's say we have to create something called hemoglobin, which is a protein in your blood. And DNA says, I do by decree state that we must create hemoglobin. And what he does is he writes out this recipe. Technically what he does is he just verbalizes the recipe, and he's got this his little RNA little servant to write it down. That's called transfer. He's transferring the ingredients onto another location. Then what happens is that now recipe and the order leaves the nucleus by a messenger. This is known as messenger RNA. And he's going to take the message out of the castle to the people who can facilitate the order. What then happens is he takes the recipe, he finds a ribosome, and this is my order, and he feeds it through, and this goes, and he reads the recipe, plucks the ingredients from free amino acids, joins them together, and creates the appropriate protein. That's kind of the point of RNA. We have to get the recipe down on paper. We need to messenger it out of the nucleus, and we got to get it to somebody who can create what we need to create. That's it. That's the role of RNA. I kind of like my little mental person, that RNA. He's just this little go-getter. He's just like, you got it, boss. And off he runs. This is this super eager beaver who's just willing to do anything. Think like DNA is Michael Scott, RNA is Dwight. He's willing to do anything quickly to get it done. That's RNA.
Does the rRNA go to is it the cytoplasm?
So the rRNA, the ribosomal RNA, specifically goes to the ribosome and creates the protein. Now, depending on, in the next chapter, we're going to talk about what happens after that. Depending on what I need to do with that protein, either I'm going to keep it inside the cell and use it inside the cell, or I'm going to ship it to the FedEx, which is called the Golgi apparatus. The Golgi is going to repackage it and then ship it to the phospholipid bilayer and release it out to the environment from the cell. So it kind of depends. Do we need to keep the thing in the cell, or do we need to ship it outside of the cell? And those are two different processes that we would use. But as far as our rRNA, that is specific for ribosomal, and the ribosomes are what make the proteins based on the instructions they are given from the king DNA. So here we have just a picture. Here we have our double helix DNA. And basically what's happened is we're going to say, okay, this one stretch of DNA, this tiny little segment, itty bitty segment of DNA, this happens to contain the ingredients that we need to make the protein. But I can't snip it off. I can't damage the DNA. So what I'm going to do is I'm just going to copy half of it, because I already know, can I create an entire structure from one half of DNA? Of course I can. We did that yesterday. That's beginner stuff. So when I've copied it down and I now have this half, that's now our RNA. Here's our little picture. I love this little picture. Get my pointer here. So here we have the castle. This is the castle. That's the nucleus. Here's King DNA, and he's about to send a proclamation. Here he is. And so what he's doing is he's copying part of himself, and that's going to be our tRNA, or translation RNA. Now we're going to messenger it. We got to get it out of the castle. So we boop, 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 messenger it out. We're going to find a ribosome that can help us, and we're going to feed the instructions through the ribosome, and we're going to make the protein. So we've gone from tRNA to mRNA to rRNA.
So tRNA is just like the copy?
Mm-hmm. That's it. To transcribe means to make a copy. Whenever I think of the word transcribe, I think of like the stenographer in the court who's writing down every single word. And when they're like, can you read that back? And they literally have every single word written down. That's transcription.
So is the ribosome itself RNA, or does it contain RNA?
Neither. So what ribosomes are factories. They are worker bees, but they have to be told, what are we making today? Because ribosomes can make any kind of protein, literally any protein. So what they do is they wait for the specific recipe. So the rRNA, the ribosomal RNA, is the recipe. These ribosomes are encouraged to come down and read the recipe. And by reading the recipe, they are then able to make the protein. So the RNA and the ribosome are two completely separate structures that work together to make the protein. If we want to think about it in a slightly different way, I think, Amanda, was it you that asked the question? I didn't see. Yeah.
Yes, that was me.
Okay. So I am the ribosomal RNA, and I have decided, my king has decided we need to bake. I'm going to go off proteins for a second, although ribosomes only make proteins, but I need to make a cake. I need to have somebody bake me a cake, but it has to be a very specific cake. And so I have the recipe, the ingredients, but I can't do this myself. All I got is this. So what I'm going to do is I'm going to come to you. You are now the baker and the ribosome. And I come to you with my transcription that I have messengered. And I say to you, hello, ribosome, I need you to make me this cake. And you look at this and you go, sure thing. Now, the difference is, this is where it's going to go into bizarro land. You basically fold this up and you go, and cake starts spitting out of your head.
So I guess my follow-up to that is, does the same strand of RNA change whether it's tRNA or rRNA, or does it copy multiple times?
It kind of depends on how many proteins, but when we're talking about T, M, and R, what we're talking about is, so this is the RNA that is transcribed, and tRNA, that's telling us what stage are we in, we're writing down the recipe. Then this RNA is released and moves out of the nucleus. We now refer to this as messenger. So this strand of RNA is the clipped off strand from here. Here, tRNA is just, all it's telling us is we are making a copy. We snip off the copy and it drifts as a messenger. And then this RNA, this ribosomal, is the same RNA from right here. But what we're referring to, our RNA, is now we know it has found the baker and it has given the instructions and we are making the protein. So it's the same RNA, just telling you where in the process. Have I got my copy? Have I messengered it out of the castle? Have I delivered it to you to spew cake out of your head? Same piece of paper.
Miss Huff, my phone is glitching and it keeps throwing me out of my Zoom call and throwing me back in and throwing me out and throwing me back in.
Okay, well, I will have this lecture posted to my YouTube channel later this afternoon, so that you can review it, because we just did a fabulous story about spewing cake out of your head.
Okay.
So, does that answer your question, Amanda?
Yes, absolutely. Thank you.
Absolutely. Did I lose anybody on the bus with the head cake spewing analogy?
Nope, it's like a copy machine.
It's you know, that would have been a much more rational way to describe it, but I like my visual of cake spewing out of your head.
No, the cake visual got me on the bus, so we're good.
Okay,
Yeah.
good. So guys, sometimes this stuff just literally pops into my head and I'm like, hey, let's see where this will take us. Here's another picture. Here we are making our copy. This is our tRNA. We're messengering it out, mRNA. We find the ribosome that can help us. He makes the protein, rRNA. Now, what do we need to do all of this? Well, we need energy. and I know those of you that have taken some sort of biological or organic chemistry class have heard these three letters until you are sick of them. ATP, adenosine triphosphate. This is how cells work. This is the energy of our cells. This is how they provide themselves with fuel and energy and able to do all of their jobs. Because essentially, we learned this in the beginning of this chapter. If I have things bonded together and I break the bond, what do I release?
Energy.
Energy. So what happens is I have three, we'll call it like here, three little phosphorus molecules bonded together. ATP. One of them, pew, breaks the bond, releases energy. I now have ADP, adenosine di-2. I can't move my fingers. 2. Then I come back and I rebond. I break the bond, I rebond, I break. Every time I break that bond, pew, I release a little bit of energy. That's the whole point of ATP. It just breaks a bond between the phosphorus molecules and releases the energy that the cell or the organism part of the cell needs to do its job. We're going to get into this a lot more in the next chapter.
So basically, every time ATP is turning into ADP, it's releasing energy.
Yeah, every time ATP breaks a bond and becomes ADP, it releases energy. Then we reform that phosphorus bond, break it again, release more energy. So ATP converting to ADP, reforming into ATP, breaking down into ADP. It just happens over and over and over and over. And every time it breaks, it releases the energy.
Ms. Huff, in the test yourself 212, it says, how does the ATP molecule differ from a nucleotide? I put, it's probably the complete wrong answer, but I said the nucleotide has a single grouping of phosphate groups, and the ATP has 3.
Mm-hmm.
So would that be the proper answer to that? Is that the difference?
Yeah, that would be a great answer. Another way that ATP differs from our nucleotides is that the nucleotides forming either DNA or RNA, they're not going to essentially, they're not going to break the bond like ATP does. When we have a DNA writing out the instructions becoming RNA, we don't have the same energy release because we're not repeatedly breaking a bond. Where ATP can break and reform the bonds pretty much until it's spent. So I would say that DNA and RNA is far more, the structure is far more finite. ATP is far more able to manipulate itself as far as releasing and gaining a molecule.
And ATP is only with RNA. Correct.
No, ATP is the functional energy of cells and cell parts. ATP is how your cells do their job, how they have the energy to do anything.
Okay, thank you.
So this is important, and we're going to talk a lot more about these bonds because there are things like drugs, chemical reactions that can slow down or speed up the breaking of those bonds. So we can slow down and minimize the amount of energy our body and cells get, or we can speed it up and maximize it. We can direct the energy into other areas. We have a whole lot of things that can manipulate the ability to break these phosphorus bonds. And so you'll learn a lot about that in things like pharmacology and clinical pathology. But it really does also help us understand why and how cells work the way they do. Are they high energy needing cells? Are they low energy needing cells? And kind of that will give us the idea of how to best support that system of the animal. And that is all she wrote.
What did you say the difference between ATP and the nucleotide was?
Okay, so ATP is the energy source for the cells, and their bonds are much more easily broken and reformed. Nucleotides are the proteins that make up DNA, and once the bonds are formed, they really don't break.
Do they release energy when when ADP is reforming back into ATP? Do they also
No.
release energy?
They only
just when they're
rele-
breaking.
They only release energy when the bond breaks. And I understand I'm cutting out, but I am so sorry. I will make sure that I charge my hotspot. I forgot to charge my hotspot last night and it's at like 18%. So I will make sure I charge it tonight so I have it on standby for tomorrow. All right, so we've got a little bit of time left. I thought what I would like to do is just kind of do a howdy-do introduction into chapter 3. I think on the Moodle, chapter 3 is divided into 2 sections, part 1 and part 2. It's same chapter. It's just that for some reason, the photos on my PowerPoint didn't compress properly and it was too big to load as one single PowerPoint. So it kind of divided it. This chapter in your textbook is called The Amazing Cell. So we're not out. Now, none of this stuff is going to be on your test tomorrow. Only the stuff from chapter 2. So now we're out of molecules and we're now into looking at cells. Cells are the basic functional unit of all living beings. And in fact, the cells are the basic functional unit for all of your individual systems. Now, ultimately, cells kind of contain the same structures for the most part, but they can look very, very different depending on where they are in your body. For example, a muscle cell looks very different from a nerve cell. And a nerve cell looks very, very different from a kidney cell. And that's because form follows function. The function of the cell is going to determine what does it look like and how does it work. And this is great because I do not want to have kidney cells functioning like kidney cells in my heart. I don't need to be making urine in my heart. And I don't need liver cells migrating somewhere else and saying, you know, this little liver cell is like, screw you, mom and dad, I want to go and be a nerve cell. And suddenly he's like, and he becomes a nerve cell. And no, that doesn't happen. That can't happen. So cells can live alone. You don't have to have them as part of a complex structure. We are the most complex structure. But we also have single cell organisms. In fact, we've got some single cell protozoal parasites that are pretty nasty. You're going to learn about those. We've got things like spirochetes and Giardia and Clostridium and that sort of stuff. So they can live together as a complex organism, or cells can stand alone. They can be single cell organisms. Now, the thing is with all of our cells is they possess a unique genetic code. And that genetic code tells them who are they, what do they do, how do they work. It's the genetic code that tells my kidney cell, you are a kidney cell and you are going to function like a kidney cell. And this is how you do it. Heart cell, same thing. You're going to function as a heart cell and this is how you do it. So even though we can have diversity in the appearance, the cell is going to have this unique genetic code that will tell it exactly how to behave. And even though we do have this diversity, all cells ultimately have very similar structures. So in our multicellular organisms like us, as I said, we have cells that are specific for certain jobs. That's called differentiated, meaning that one group of cells will have one job, another group of cells has a differentiated job. And all of these systems have to work together in order to sustain the life. So in order for us to truly understand and comprehend how to support an animal anatomically and physiologically, we got to have at least a basic understanding of how the cells work and how they are supported. I'll give you guys a minute. So throughout this chapter 3, we're first going to talk about how do cells regulate what comes in and out. And then we're going to talk about how do the structures of the cells facilitate that to happen. So it's called the organelles, or the tiny organs of the cell. So let's talk a little bit. Cells are really pretty amazing. They've been evolving for billions of years. Now, this is where it gets kind of cool. So in our original evolution of cells, we had these little, they weren't even really cells at that point. They did not contain the organelles. They did not contain the structures that we know for our cells today. But our primitive cells had a single strand of DNA. And remember, DNA is just the instruction manual. You are this kind of cell, and how are you going to behave? So
no nucleus.
They did not have a nucleus. They didn't need one. And so we have our prokaryotes, pro meaning before. These are our ultimate super primitive cells, did not have a nucleus. Because the structure of the cell was so primitive, I did not need to move and translate RNA. The DNA was able to govern just by being proud. And so our DNA was not separated. Now, as these cells evolved and became more and more complex, suddenly now we formed the castle, we formed the nucleus, and this is where our DNA is going to be housed. So prokaryote means, before nucleus. We still had DNA, but it was not in a nucleus. And then as our cells evolved and became more and more complex, suddenly now we have developed a nucleus to separate the DNA and be able to govern this more complex organization of organelles. Now, kind of the cool thing, so I find this kind of fascinating. So we've got, I'm pretty sure all of you know, if I asked you, here's the question everybody asks. What is the powerhouse of the cell?
The nucleus? Or no?
Mitochondria.
The mitochondria is the powerhouse of the cell. The mitochondria is what gives the cell ultimately the power it needs to function. This is evolution. Pew, pew, pew. So it's thought that the mitochondria originated as a prokaryote. It was a very primitive, no nuclei cell on its own. It was an entity on its own. And basically what happened is you have these little prokaryotes and you have these other cells, these prokaryotes that are now evolving and becoming more and more complex. And
you cut out after you originated.
Oh, okay. So you have these prokaryotes, and actually, so you have these prokaryotes, And think of like a whole group of them. And there's a couple over here that are evolving and they're becoming more and more complex. And the prokaryotes are like, hey, look at what's happening with those guys. That's not happening to us. And suddenly these come think like mean girls on a cell level. They're like, ha, we're all that. We're super complex. But the problem is the more complex these cells got, the more energy they needed. And they didn't have the energy to provide what they needed to continue to become complex and to function. So they said, okay, wait a minute. I think I see something that could work. So they go over and they say, hey, prokaryote, we see that you are just over here making energy. And, we're over here evolving and becoming way more complex. But here's what I think we should do. We should team up. And you can come and be part of our super cool complex evolution group. And you can be part of us, but you have to provide and you have to make energy for us. And the mitochondria was like, hey, that sounds like a pretty sweet deal, man. And so that's exactly what happened. In theory, is that the mitochondria became incorporated into the complex cell. The mitochondria now has the benefit of the functionality of the complex cell, and the complex cell is benefiting from the energy production of the mitochondria. And the reason why we think this happened is because mitochondria contains its own DNA. It has a non-nucleated strand of DNA, just like our prokaryote. And so it's kind of fascinating how, even as a cell evolution saw the benefit and was able to incorporate and both parties kind of benefited, although obviously I anthropomorphized it, but you get the idea. And so I find that kind of fascinating how, through evolution, we can be very selective and choose things to incorporate that are ultimately going to benefit for the long haul. So I find that kind of interesting. So that's the evolution of our cells. Here's just a picture. Now, this particular, here we have our prokaryote, and here's our strand of DNA. Now, this is deceiving because this is showing that the size of our prokaryote and our eukaryote is about the same, and it is not. This prokaryote is, in fact, about the size here of this mitochondria. So that's a little bit more accurate depiction of the size difference. And these mitochondria, if the cell is like, I'm giving it all she's got, Captain, she ain't got no more, and we still need some power, mitochondria can go, on it, boss, and he can pinch himself off and make a second mitochondria that can then increase the amount of energy being produced, which is pretty cool. It's pretty awesome. And I'd like to say, little Star Trek nod right there, with the most horrible Scotty accent on the
Yeah, I totally
planet.
recognized that.
Yeah, it was the worst Scotty accent, the worst Scotty impression in the history of doing Scotty impressions. So I got that going for me. So why are cells so small? Why don't we have these boulder sized cells that can just, function on their own? Well, the problem is the power and the nutrition and the energy. Smaller cells are more efficient. They are more efficient in carrying out their functions. Larger cells would not be able to take in nutrients, and we would not be able to provide the energy needed. Now, is that to say we don't have some extremely large cells? Of course not. Nerve cells are some of the largest cells in your body. But the other thing is that the larger a cell is, the more kings you need to govern it. Think of the Game of Thrones. Game of Thrones. Think of the whole world as a cell. The 7 kingdoms together form a cell. But the kingdom, the 7 kingdoms, is so vast and so large that each section needed to have their own ruler. And each ruler lived in a castle, the nucleus. So when we have super large cells, we have to have multiple kingdoms, multiple castles, multiple kings and queens to rule over it. Now, just like in, okay, spoiler alert, just like in Game of Thrones, the more kings and queens you have, the more potential problems you have, because everybody may be trying to vie for power. And so we see a lot more cellular complications with these complex multinucleated cells than we do with a single nucleus. When there's only one king, there's nobody there to say no. If we have many kings, suddenly now the line of communication can be broken at multiple places. And so that's kind of the difference between our very large, complex, multinucleated and our very small, single nucleated. Does that make sense to everybody? Did everyone get that game? I'm throwing out a lot of analogies today. I don't know why. Today is like Analogy Wednesday. I've gone from cake spewing out of your head to Star Trek to Game of Thrones. I'm pretty proud of this progression. Any comments, questions, concerns so far? All right, so every cell has the same basic essential anatomy. We have the cell membrane. Now, the cell membrane ultimately is what governs what comes in and out of the cell. We don't want it to be open general admission. We don't want everything having access to the inside of our cells. And so we have this cell membrane. I think of it kind of like the skin of the cell. Just like our skin serves to protect us and regulates what goes in and out, same with our cell membrane. Sometimes we also call it the plasma membrane. And it is the structure of this membrane that helps to regulate and keep some stuff in and allow some stuff out and allow some stuff in, but to keep most of it out. Now, inside the cell is kind of this liquid jelly. It's called cytoplasm. Cyto meaning cell, plasm meaning fluid or fluid-like structure. And so everything is suspended in the cell in this jelly-like structure. Think of it like, I don't know why, but Thanksgiving, my mother always makes carrots in Jell-O. I don't know if any of your moms ever made that. It's Jell-O that has carrots mixed into it. And it's bizarre. And nobody eats it ever. And she makes it every year. I don't know why.
That poor Jell-O.
I know, it's disgusting. It's so gross. It's so gross. But if you think the carrots suspended in the Jell-O, that's kind of what your organelles are doing inside the cytoplasm. And the cytoplasm itself, though, unlike Jell-O, is super, super complicated, super, super complex. The cell, when we think of a cell, when we look at a cell, we usually will just draw a circle and we'll draw the nucleus. Holy smokes, nuh-uh. I got a little video I'm going to show you probably tomorrow of just how complex and complicated these cells are. We have a whole internal skeleton that helps to maintain the shape. And then we get a whole secondary skeleton that helps the cell to change its shape. We have guide wires that the organs can attach to and move around. And so the cytoplasm, super, super complicated. And then, of course, the third and final structure is the nucleus, which contains our genetic material. So our cell membrane is the skin of the cell. Our cytoplasm is this jelly-like substance, literally everything inside other than the nucleus, which is right here and is a separate structure. So here we have our cross section. So here's the jelly-like cytoplasm, this gray. These are all of the organelles. Absolutely. We have organelles that are suspended, but we also have, as I said, a cytoskeleton. And so they can like grab on and move around this cytoskeleton, just like pulling themselves along, because there may be areas of the cell that you know needs more organelles than another area, or a part of a cell that needs more energy than another area. And so the mitochondria just and they look like giant slugs, and they'll go and provide the energy that is needed for that other part of the cell.
I'm good now, Rita.
So here's all of our organelles. We're going to talk about all of these functions. So there's the one we know, mitochondria. This is a Golgi body, ribosomes. We have some ribosomal RNA right here, or some endoplasmic reticulum. Here's some smooth ER. And we're going to talk about all of the functions of each of these in the next part of the chapter 3. All right, so time flies when we are having fun. All right. So, what questions, comments, or concerns do you have? Am I going too fast? I'm seeing some comments that I seem to be going too fast.
Sometimes you go click through the slides fast, but
Oh my goodness.
I just go
Please,
back.
please, 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 please. If I'm going too fast, you stop me and tell me immediately. Because guys, actually I'm gonna pause this.